Excellent, thank you. So yeah, that family, the Chrysogorgia of octocorals, is characterized by those kind of thin, elongated polyps. Has a very different look. And what's interesting about this ridge is, again, I mentioned it's going from the west to east direction. One side of that ridge, the one that we're on diving on, so the kind of southern portion of that ridge has a very steep drop off, and that would be to our right. On the other side of the ridge, it's a lot more gradual. So we assumed that this would be a better dive site because currents would be accelerated over it. And then we have seen this kind of flow from right to left right over that feature. So yeah, there's a little bit of discussion in the chat room about collections, and yeah, I absolutely agree with you, Chris. So if there is something that is I assume most of these things will be completely new, but
I'd suggest if we see that Chrysogorgia again, that was really interesting looking. That would be a candidate.
Copy that.
Hey, Washley, we're going to do a quick pilot change, so just please stand by.
Copy that. Yeah, so we're just going through a quick of a personnel change here, our piloting team. It's lunchtime here in the ship, so our engineers and pilots are going to rotate through, and so just stand by for a couple minutes. Again, this is the 8th dive of a 3-week expedition throughout the northwestern Hawaiian Islands, including work both inside the Papahanaumokuakea Marine National Monument and the surrounding waters. Unfortunately, this is also the last dive of this expedition. Unfortunately, tonight, we have to depart for Kwajalein Atoll, where this expedition will end next week. But fortunately, this is only the first of 4 cruises that the Okeanos Explorer is going to embark on this field season, doing work in some of the most special places in the Pacific, some of the largest, most remote, and most beautiful marine protected areas in the Pacific. So after this, the ship is going to Wake Atoll and then to the Marianas Trench, and then coming back through the Pacific remote islands before ending the field season in September back in Honolulu. And then, of course, that's luckily only the second year of a 3-year effort by the Okeanos Explorer. So next year there will be additional work throughout the U.S. Pacific Islands. And you can tune in, follow along, and follow these expeditions online at oceanexplorer.noaa.gov. Of course, updated information about these cruises will be posted, and then, of course, you can also follow the video in real time over the internet.
Okay, how we doing, Watch Lead?
Excellent. Yeah, we'll keep our eyes open for some collectibles. So there are pretty much, there's a lot of things that are new, but yeah, we'll cover a little bit of ground, and we'll point out things that look like we need to image them.
Okay. Co-pilot, which I'm just heading up the ridge, and you're facing
yeah, I'm facing that direction.
Okay.
And we haven't been moving. It's about a 15-minute delay, he said.
Okay. Yeah, and you put it in a move about...
So I haven't looked at the sonars.
13 minutes ago. Okay.
I assume that sonar is not alarming, right? It's just... that's what a ridge looks like if it's not too steep.
Yeah, it's not very steep.
Given that we've got a lot of stuff behind.
Yeah.
Do you want to go ahead and start a move?
Well, I think he called in a move about 12 minutes ago.
Oh, really?
Yep,
Okay.
I expect Sirius to start moving in about 2 minutes, maybe even 1 minute. And it's going to move 30 meters.
Okay.
I estimate like a 15-minute layback. So, yeah.
So, pilot, do you want me to keep you centered up, or do you want me to stay pointing the way we're going?
Well, what direction was that move?
085.
0.5. Okay.
That's towards waypoint 2.
Okay. Yeah, you're looking down at 50 degrees, so I'll just push out ahead a bit.
And
we're
yeah,
not in a rush, so-
Scott Franz from the University of Louisiana just provided some
So
important
I'm on
information
085 now,
that
so I'll hold heading
Chrysogorgia,
and
that thin
just
wispy
tilt up
coral
to keep in.
Yep.
that we imaged a
Sounds
little
good.
bit
And rise up, I guess.
earlier
We're down to
looks
pretty
like
small depths, aren't we?
Chrysogorgia pinnata.
Yeah, we'll want to come up
And
a
that
bit.
was imaged
And
at
video,
depths
are we
of
full wide?
3,100 meters in Pacific Seamounts, in the East Pacific.
Okay, now I'm at the end of my tether here. So I'll hang
Oh,
out here.
I'll
All right.
Stop coming up.
All right, watch lead. Is there anything that you see in the area, or anything in particular we're looking for?
Yeah, let's maybe the sponge dead ahead. I don't know if it's too far away. We haven't gotten a close up, but if
I'm
not,
at the
there's
end
a
of my tether, but I can
there's,
set down and get it for you.
there's a coral also to our right that looks yellowish, a little bit different. So it's out of view now. If that's too far away.
We can do a quick zoom here.
Any chance we could look at that little sponge on the left, the little one on the little bulb on a stalk?
Yep. So if you heard that, Dan, there's a little sponge to our left, right on the bottom left corner.
yeah.
Just got in view now.
Do you want to do a quick zoom on this stock?
No, I think the
No?
one to our left is
Okay.
a bit more interesting, given that it's.
All right.
There's another one of those Chrysogorgids right there too.
Excellent. So yeah, I'll put that up for discussion. There's a little bit of discussion about collecting that, and I'll defer to some of our shore-based scientists. It certainly is different than
closer.
anything that we've imaged here in the region, so it would qualify for that. But so, if any folks have some opinions about that, it definitely does fit the category of collectibles.
Okay, zoom in video.
Okay, well that guy's different.
Yeah, very unusual. It doesn't look like I don't have it here in our guide. I was hoping that we would.
No, it's not in the Hurl Guide either. It's probably in the Bolosomini. It's probably a euplectellid, but I don't know. That's a guess.
Excellent.
All right.
So,
Are we interested in this one, or?
well, I think there was a consensus, perhaps, about there's a coral, a wispy coral
Okay.
to our left,
Let's zoom out. Thank you.
and so it's about a meter left and down.
So are we pretty confident we want to collect here?
I believe so. Yeah.
Video switching
Then
out.
we'll want to.
Yeah,
Stop
we
the
could
ship.
stop the ship.
It's probably
It's probably close anyway, but
we're going to be moving regardless.
Yeah.
yeah.
Okay. Is it this
Bridge
one
now.
here, just below the lasers? to
Yep.
the-
But it's only
Exactly.
30 meter move, so
If
Oh,
we're not
Okay.
done that move, could we bring it to an easy stop? Thank you. Copy, Bridge.
Okay, but it's only 30 meters. So we
yeah.
should-
Yeah, it was only 30 meters,
I
and
think I'm starting to move.
I'll keep an eye on that horizontal marker in your
Okay.
sonar.
Are you wanting to zoom on that pilot?
All right. So yeah, there's
Yep, let's zoom in.
seems like we do have a consensus here, and we're going to go set up and collect this very unusual Chrysogorgid coral.
Okay.
So current depth is 3,963 meters. We started
Alright,
to dive about
that's
a
fine.
little over an hour ago. We're diving on a seamount that's completely new. It was mapped for the very first time in the early hours this morning. In fact, they just finished mapping it at 4:30 a.m. this morning.
Okay,
And,
roll. Let's zoom out, and we're going to collect.
and when all of us woke up and saw the maps and the complexity of the topography, we were all
You're gonna need to
very,
talk me
very
through the hydraulics.
surprised
I can
and
help you now.
intrigued. And
If you want to
we were just
ROV
trying
hydraulics,
to pick up a site,
Yeah.
and so there was
and
multiple ridges
hydraulics are on tab to the
to
right.
pick from,
Yeah,
and we decided to go for the
Enable.
shallowest of one of them, but that was still quite deep, 4,000 meters deep, and
That's a little tricky.
we did not expect to
Go
see
higher,
as much
maybe.
life.
Yeah, there you go.
And yeah, it seems like
And
we've
On the right, drag that slider up.
come across a nice
Keep
moderate
going,
density of
Keep going right there.
corals,
Is
sponges. Also seen
good.
a
And if
couple
you,
of sea cucumbers.
I would just turn
And
on
right
both
now we're
valve
just,
packs. So
setting up to collect our
down there, if
first
you got a port
biological
valve pack, and
specimen
then
of
hit
this
enable.
dive. This is a unidentified
starboard,
species of
give him
Chrysogorgia, in the
craft
genus
control.
Chrysogorgia.
and that way we can use either wing if you need to, port or starboard. So
Okay, seems pretty stable.
And
to me.
the only guess here
So-
for this species is Chrysogorgia
Where do you wanna
pinnata,
go
which
with
has
this?
not
I
been
guess we should make
identified
sure that
from
we don't have
Hawaii.
any other samples.
well, we only have one geo sample, and
One
that is in
geosample?
starboard
Yeah, let's go with the port
rock box. So let's do port
outer,
bio box.
port outer bio. Yeah.
Okay.
I guess one of the questions I'd have is, because the structure is so simple, is just the image itself strong enough to identify it as pinnata and say that it's a new record for Hawaii?
Come up half a meter.
Yeah, I'll defer to you folks.
And then
I don't
extend
know
it,
this
put it
group well enough,
Feel comfortable doing that?
Yeah.
but we'll have to make a decision quick.
Chris, if you
So how will I
posed that
extend it? Will I be
question
able
to Stephen Cairns, he would laugh at you and
we
say
don't wanna
that
do
he
it
cannot
yet.
No, I
identify
know,
anything
it's
from
on the,
photos without a
I
specimen.
believe,
So
starboard
if
Yeah.
you were
drawer.
here,
Yep.
Open close.
that he would
Port-
advocate collecting
port
it.
bio retract extend?
Also,
Right,
if
so
you
the-
guys want or
the
need
procedure
a
is,
better image of something
so
on
yeah,
the
close
thanks.
the
coral,
drawer all the way.
you can just let
which
us
you
Thanks
can
for
go ahead and
providing
do.
that, Scott. So
Right
yeah, I
now.
think we'll go
Close
ahead
the
and
drawer all
collect
the
this
way.
one.
Which is
Retract. We
no,
talked
the
Dan,
drawer.
one of the things
so we
is
wanna
that
bring the drawer. It tends to
pinnata
creep out.
has second and
Close.
third order
So
branches,
close
and this
that
one
all the way.
doesn't have that. It has the branches coming
Yeah, it
off the
looked like
main
it came
axis
in a little bit.
Yeah.
and no branches coming off the branches.
And
So
then,
that suggests, I thought maybe it was a young
and then
specimen, but
retract.
but it is. But I could see clearly that
That's
it was
starboard.
reproductive
Yeah,
because the eggs,
Okay.
the places
Oh,
that
yeah,
it stores the eggs,
Did you say
which
retract?
we sometimes call saddlebags, are clearly
Correct.
developed,
Retract.
and so it's a reproductively
And then
mature
extend.
specimen,
And
but
that
it
should
doesn't
keep the
have
lid
secondary branches.
open
So
when we open
I
the
would
drawer.
suggest from that it's probably not pinnata, but something else.
excellent. Yeah, thank you
Okay.
so
Just
much.
port.
That
We're
was
just messing
Dr.
with
Les
port.
Watling from the University of Hawaii, who another expert on
If we had a
deep sea
sample
corals
in the
and on
starboard one,
Chrysogorgia
we'd want to
in
make sure that that's
particular. So yeah, thank you very
retracted
much, Les, for
so that
providing
the
that
lid would
context.
stay
So yeah, we'll
closed
go
on it,
ahead and
but
collect
we
the
don't,
sample.
All right, are we ready to go? Hydraulics on and ramped up?
I think so. I think we're all set.
Okay.
video. Nat, could you bring the current? I don't know if you can, but the current mini Zeus view. Can you make that Sirius's view? Okay.
I'm going to put the ROV HD2 here.
So since we know that Sirius is moving, but we're confident that it's not going to
And
move
Jim, if
into
you
a.
can take control of Mini Zeus.
Dan, the other thing is
Hey,
there's
real quick
Set
before we do that, can
another
I-
Chrysogorgia is just on the edge of
Hey,
the
pilot
right-hand
and
side
co-pilot,
of the
before we
image,
do that,
and we'd like to have a zoom
We
on
know
that
that
and have a look
Sirius
and
is
see what that
moving.
is.
so I'll
Copy
keep an
that,
eye on
Les.
Sirius.
We'll get that for you.
But before we put
As
that
soon as the
camera up,
collection is
we need
completed.
to all be on the same page for Sirius moving.
Sure.
Just knowing that it's still moving.
Yeah.
So I'll keep an eye on that. But you guys do the cameras and
Okay.
everything you need.
Okay.
Yeah.
Thank
So
you.
to get the mini Zeus,
Yep.
press that. So
And then now you have control on the left
top left.
joystick there. Yep.
I'm
Thank
sorry?
you.
So now you have control on that joystick.
Okay.
So if you can bring it around right to the craft.
The other issue with this coral is that it's going to be very, very, very light. And so my recommendation is that when it's being put in the bio box, that the manipulator end with the coral in it be put all the way into the bottom of the bio box before the claws open, and help to minimize the possibility that it'll float back out.
Did you hear that?
I did not catch all of that.
So it's going to be really light.
Okay.
So we want to make sure that it's all the way at the bottom.
Before we let go.
Yeah.
Okay.
And you can also go ahead and collect it as far down as you can, yeah.
Okay.
So we can go ahead and collect.
Science, based on all the
So
discussions you guys
maybe
are
we should
having
try
on
to
this
see if
particular
the
specimen,
bio
are
box
you happy
We'll
with
open
the
you off for a second. Okay.
video that you've seen currently
All right,
of
are
it?
we
Do
ready?
we need to get anything any better?
No,
Yeah.
that was a good close-up, Roland. Thank you.
Yeah, I thought we had pretty good close-ups of the polyps and branches. Thanks, Roland.
So think about which side the coral cutters are on.
Should be on the underside, but I will check.
Yeah, you got the blades on the top right now, right?
Yeah.
Yep.
There you go.
Okay.
But if you call a zoom on any camera, I'll be standing by to
Okay.
give you a
Copy.
better view.
Thank you. Wonder if I'm too close here, actually. I mean, I'll be able to
Think
get
you're
it,
okay. I'm gonna get you in mini Zeus though.
but yeah. Yeah, we got a good shot of it. And if you could bring Mini Zeus down.
Sorry, I was looking at the wrong coral.
It's a good perspective there.
Thanks. Should have set up a little further away, but I'll be able to get it.
I think you're okay there.
Oh.
Yeah,
That's what I
your
did.
wrist is hitting there.
Yeah.
That's good.
Yeah, excellent grab. And then just for folks to give you a reference, those two lasers on screen, those are about 4 inches apart, 10 centimeters.
Is that a good position to then pull off?
So that gives
Yeah.
you an
Step.
indication
Yeah, I
of
wouldn't
size
move
of
it
some
much
of these
more.
things. But even with that,
The more you
sometimes
move it,
we're
the more likely it is
surprised
to
when we
wiggle.
Sure.
when we actually measure some of these colonies, and
Alright.
you might have seen some
So
of
we'll
these
come up,
enormous
put it on a
stocked
deck.
sponges, and there was a few unbranched bamboo corals earlier that
It's nice
looked
and
like they were as
slow.
tall as the vehicle. And yeah, it's always
Hey, Daniel, it looks like there's a swimming polychaete coming here into view.
yeah. We got to collect finish the collection first, and
That should be plenty.
Just oscillating a bit.
Yeah, it's gonna
yeah,
oscillate a little.
he's not shy of that polychaete.
You can change your set point a little.
Polychaetes are commonly known as the bristle worms, and it's also a very species-rich group.
Not OXY, so don't drift back.
Yeah.
I believe close to, or if not
Should be
more than
perfect.
10,000
Okay.
species
I'd go ahead and,
are known.
Yeah,
And
let's
get the drawer out.
we can get the drawer out.
they
So
get their
extend
main polychaete.
open
Chaete,
drawer, right?
are
And
those
you wanna
bristles.
make sure that
that's
the,
those kind of hair-like projections they have
box-
from
We
the
did.
side of their
we
body.
Did. We did.
Yeah.
And so polychaete means literally many bristles, and so that gives the other common name of the bristle worms.
Cool. And then let's bring the mini Zeus.
And a very, very
You
common
can
and
follow me
abundant
with
group.
it. And let's
But they're small.
get a.
I seem to have lost. Oh.
So yeah, one from a biological point of view, one very interesting thing about this expedition in particular. So we've been targeting particular habitat features where we think we're going to find higher abundances of animals. So we've been going to these.
Ready for drawer close?
Nicely done.
Outstanding
Yep,
job
let's
by
close it
our
up.
pilot. Well done.
Nice.
Okay. And as I bring the arm back in, if you can follow me on mini Zeus.
Yep.
Great. Make sure all my joints are.
Yeah, very well done, and you're getting all kinds of compliments here, Dan, in the chat room.
Thank you.
Spec O2 Bio.
Follow me back a little more so I can see the shoulder. Thank you.
So as soon as we're all situated, there's a request to zoom. I think there's a coral just right of the screen, center right. Once we're all stowed away.
Sure.
Yes, I was saying that one of the primary themes of this expedition and the previous one is to look for high-density communities.
How's
So
that
we're
look, Chris?
looking for
Good deal?
places where
Looks
we think
good.
we're going to find them, but we also did a couple dives,
How's that looks coming off?
including this one and one just a couple
Nice.
days ago where
Thank
we thought they might not be so high, and that was today because of the extreme depths. And then a couple of days ago when we dove
So
right through the oxygen minimum layer,
before we
and in
turn
both of
hydraulics
those
off,
cases we are
would,
seeing fairly moderate densities.
Retract that pin.
So that's quite surprising
There
to
you
some
go.
of us.
And then extend it.
That
Well, we
No.
want to make sure if we open the
No.
drawer again that it
Yeah.
doesn't
Yeah.
open.
Got it. Got it. Okay.
So,
And then turn hydraulics off.
well, you can disable both valve packs, and then I think
but
we're
yeah,
clear.
for those folks who
to,
joined us on
spin down.
yesterday's dive, that was absolutely spectacular. We
On
were
our
on a
heels,
ridge
then, yeah.
around 3,000 meters, about 80 miles north of here,
And then we just
on
disable.
another,
We keep it on, just disable it.
seamount, and there was corals and
Yep.
sponges as
Perfect.
far as you could see, and you couldn't even
Nice work.
find places to put the vehicle down in some places,
mentoring.
just absolutely covered with life. And it's absolutely remarkable to see how much life there
I
is
think
at
before
some
you
of
go
these,
too far down, they
deep
wanted
So
water
was it
habitats.
this coral in the middle here above the sponge?
No, it's to the right and low of the lasers. It's very wispy. It's yeah,
It's below the lasers now.
yeah, just
Okay.
right at your toes.
Sure. Coming down. Okay, let's come in video. And I'll try to get it centered up here. Okay, go in further.
yeah, definitely another Chrysogorgia, a bit more complex, the branching, but also somewhat planar. Any thoughts on that one, Les?
Max zoom.
We're still collating.
Just slowly rotate around.
Yep, trying to. Just make sure I don't lose my footing.
So yeah, this family of the Chrysogorgia day, characterized by that polyp and very, very thin and elongate, and it's excellent. Thanks.
All right, let's zoom out video.
We need to see the base if we can.
Yeah, so the base, there you go. If you can zoom in on the base.
Okay, sure.
Wispy Gorgia. That's it.
Do you think that's a Plurigorgia?
Well,
What depth were these from?
these are from 2 to 3,000 ones we found, 2,200 or so.
Yeah,
That,
maybe that's what it is. Huh?
select here.
Yeah, there's another view again, kind of lined up on. So then
Yeah, those are big ones. This is just a little one. Maybe like
Just
Yeah,
hold
there's
for about
another-
a second
oh,
and
yeah.
release.
Yeah.
Got it.
Yeah,
Is anyone offer that as a possibility yet?
Yeah. There.
You put it in there somewhere?
Yeah, so I think we're thinking that it's probably a young Plurigorgia.
Is that a good enough view?
Yep,
Dan?
perfect.
Okay.
Thank you.
Okay, let's get the full specimen now.
Zoom out.
Thanks, Les.
Tilt up a bit.
So yeah, those are all of our fellow shore-based scientists that are at the Exploration Command Center at the University of Hawaii. We have Dr. Chris Kelly, a specialist in many different
Pantail
groups
controls
of
are different
deep-sea
than anything
corals
else, or
and
reversed up and down
sponges
from
and
what
other
I'm used to.
deep-sea animals, less
Some
wattling.
of them are pilot and some of them
Specializing on different groups of deep-sea corals, and
Okay,
Andrea
zoom out.
Quattrini also with a wide wealth
Airplane
of
mode,
knowledge
not
of different
airplane mode.
groups, and also
Yeah,
Jeff Drazen there,
that makes sense for the vertical.
specializing on deep-sea
Maybe, but-
fish and different groups.
Video's
So, yeah,
clear.
it's a very
Full
Okay,
wide.
thank you. Coming up.
knowledgeable group that we have here in Hawaii.
All right, so ship is probably settled out now, huh?
Definitely.
And also at the University of Hawaii
Our
is
last move
my
was
good friend Brian
Sirius is on
Boston.
that
He
head.
specializes in
Sirius.
marine geology as well. However, he
085,
specializes in
but
a
although it's gotten-
subfield of geology
it's
called seismology.
little
So
Pulled a little,
he-
but more or
he's
less.
interested in
Do we
what
want another move?
geological structures are under the surface
watch
of
lead,
the
pilot.
seafloor. Very interesting stuff too.
Do we want to hang out here and look at a few more things,
Well,
or
then
should we
let's
get the ship moving?
So, Jonathan, you need to prompt Brian to talk some more on the audio there. I've been trying to get him to do it. He's pretty quiet.
Yeah, you know, that was my my little plug for Brian to hop right in. Come
Let's
on,
do
Brian,
another
get your feet wet.
30-meter move, 085. How does that sound?
I thought everyone was wet down this
All
depth.
right, that's good. Yeah.
Oh, that could be a Bathagorgia, Bathapathes directly below us.
Can we do a ship move, please? Range 30 meters.
So it's just
bearing
above the sponge that
085
we
degrees,
mentioned earlier.
Speed 0.2 knots.
Oh, can I get my camera back?
I think I gave it back. Oh,
Good copy.
Yeah,
Thank you,
so
Bridget.
it looked like we saw our first black coral. Tina apparently had spotted one earlier, but
Yeah, Daniel, there is a request in the chat log to zoom in on that Bathapathes.
yeah, we already
Potential
called
Bathapathes.
it in, coming right up.
Let's do a quick zoom. Check it out, and if we want to get
Thank
closer.
you.
I believe it may be the one on the upper right. I'm not sure.
It's not this one.
Yeah, it's to the right of the sponge, so about keep coming. The base of it is just coming in view top.
Oh, up there.
Yep.
I can certainly get closer if you need to.
Got time.
So yeah, opposite of branching, so and
There's
very
also
wispy,
something at the
and
bottom of the screen. Did you see that
so
white?
it's definitely a black coral in the
Flowing
genus Bathapathes.
Would you like me to get closer?
Copy, Bridget.
Yeah, if you have a second.
Sure. Yeah, we got time. Let's zoom out all the way. zoom in.
So yeah, you had that very, very fine branching and opposite branching.
You have a fault on your thruster. Don't try to reset it now.
So that looks like to compare with Patula. Yeah.
Thanks, Chris.
You have to stand it up for me to zoom more.
Hey, Daniel, I have a question for you on these Bathopathes. What is the depth range for this species, Patula? Curious.
I think Tina is probably in the chat room, might be better equipped to answer the depth range of that particular species. But yeah, there's Bathopathes have been deeper than what we are now. But this is a group that's going through a lot of revisions and just a few species described, but we think there's a whole bunch that need to be described. And yeah, it's a group that there's a lot of work to be done.
Let's zoom out. I need to fix the thruster here.
Yeah.
So I'll come up a bit.
Yeah, I would come up and then
just like a meter, half a meter.
No, that's good.
So
there,
to get back to your question, Andrea, a lot of that is problematic because most of those species were described by specimens collected by the Challenger expedition and one of these early oceanographic expeditions where they collected with trawls. The samples were in many cases quite abused, and you couldn't see all of the features. And in many cases, the holotype isn't that informative. And so we've had to go back and try to see if we can re-describe some of those species so we can actually define the boundaries of those species. So it's very problematic group, and I'm sure that's the same case for other groups of corals. So for the general listeners, when a new species is described, that includes both a sample that's then deposited in a museum or a series of samples.
Or move on?
I think we'll move on,
Okay.
If we see a few of them, then we might consider a collection, but since we're already in a move. So a new species, you have a sample or series of samples that deposit in a museum, and then a description, so a paper, a text, photos.
That last move is 0.8.
And a lot of the earlier descriptions were done with specimens that were quite
you mean on that heading,
abused, collected with trawls, so you had no idea what these things looked like in their natural environment, what colors.
You had it with 1 foot
a lot of times you didn't even have tissues
really
anymore.
So you did a
samples
good job
would come back without polyps, and so you didn't really. When you want to confirm that something is that species, you go back to the description, to the text, and then if that's not informative enough, you go pull the specimens from the museums. And for a lot of these groups, those specimens aren't that helpful because they are, as I mentioned, abused. And so what we've had to do for a lot of these is re-describe the original species, try to collect samples of the same thing that we think is the same thing that was described a long time ago, and then amend the description, so add characters. And this is why this particular project is so valuable, is getting these in-situ images and video of these animals, taking a close look at how these things look like in their natural environment.
Going from south to north?
What are the colors? How big are the polyps? Any kind of ecological information we might need. Are they abundant in the environment? So all those things can be added to the species descriptions because all those things help you in identifying it.
To the right,
Andrea pointed out the depth ranges. Where is it found? That's also something we look at. Is what has been described from the region
for
before.
Series to be blown 10 degrees off.
So that gives you a starting point to try to get an identification. This is true for many of these deep water crews.
Still struggling a little bit.
There's one of the. Let's get a quick look at this tumbling, and I believe that's one of these.
Left went off screen.
Very interesting.
Hold on, Roland. Sorry, just takes a second.
Mollusk.
Change direction.
We saw a whole bunch of those last year on our deep dives.
I missed it. Sorry, watch lead. What did you want to?
Now there's a tumbling mollusk, and I believe
I don't
that's.
see him in the manipulator cam. I think he's gone.
You got room to come back a little bit,
Kind of looks like there might even be some in-place pillow lava flows right there. That big bulb kind of looks like a nice pillow lava toe, and the one next to it looks like a little small pillow lava breakout from the larger lobe on the center of the screen. Or vice versa, the bigger one could be a pillow lava coming off of the one on the right.
Yeah, and what I just pointed out, so that gastropod looked like something we identified as Gaza Daedala last year. And these gastropod, we had kind of golden shell that we saw in several of our dives, and they were always tumbling down and moving this very complex
Don't forget you switched my screen, Roland, so it's
behavior.
I don't think we have it up anymore.
You want me to put it back?
Yeah, that's fine. Thank you.
Switch it or no?
Yeah, go ahead.
Okay.
Thank you, video.
So I don't know which.
Because otherwise he doesn't have
perhaps
view.
a quick zoom in on that coral on the bottom.
No, you still
not the unbranched one, the one that's right next to it.
Okay, the tall one.
Yeah, another small bushy column right next to the tall one.
Got it.
Video's working on it for you, co-pilot.
Yep, I accidentally put it up here on the right.
Oh, okay. So you want it centered?
Front of the vehicle.
So Nav, if I select pan and tilt select over here?
Yeah, before we do that, we should come up on Sirius a little. Okay. Slant range right to the right is the...
Yeah, I'll get that in a second.
Zoom in, and I'll try to hold it steady here.
Partial.
Coming up on Sirius.
The antenna is already identified as a primnoid coral.
Excellent.
Excellent, thank you.
So Nav, I lost track. We do have a move dialed in?
We're at the end of that.
Okay.
Move. We can put another one in.
Quick, yeah, there's the lasers, and that gives you, that's about 30 centimeters high or so.
We're going to make our waypoint by the end of the day if we...
I think our waypoint is generally uphill.
Yeah, exactly. So we're just going to try to go up this ridge.
Coming up a bit, Jim.
And then
push forward.
Okay, coming up with you.
Creep towards the southern side of the ridge.
Okay. But I think maybe we should bring your heading, Jim, maybe 10 degrees to the right.
Can you brighten it up a bit, Roland?
Okay.
Just doing that.
Just looking at your sonar, I don't know what you're...
Yeah, we're going along this ridge that's on the top of the seamount, and we're going to try to... The summit is...
forcing it around to 095 or
70 to 80 meters shallower than we are. There's not really slopey. This is kind of
something like that.
More of a plateau here at the top. But this ridge on our right, on the southern side, is quite steep.
Yeah, I think that's.
And on the other side, on the northern side, it looks like more square, a lot more gradual.
Although it looks like the farther part of the ridge is going up more to the left.
That's true.
So I think we're looking at this little nose here, but by the time we move 10 meters, we'll be back to the easterly.
Okay, so let's just keep it 085.
Anything of interest here?
Yeah.
Perhaps maybe the crinoid on that coral if we're hanging out.
Yeah,
I'll put in another move. Bridge now.
Daniel, if it's not too late, if we could look at that
last move.
just going out of frame to the left.
Meters, bearing
Copy
085,
Scott.
speed 0.2.
So yeah.
I just want to see if this is like the possible Panada we were discussing earlier, because this one does look like maybe there's secondary branching.
Copy that, Scott.
Okay, let's zoom in, Roland. Kind of cool with the dark background.
Beautiful image here of this feather star or unstocked crinoid.
This is a really interesting image, Daniel, and I wonder if the crinoid landed and irritated the polyps there, and that's why there's no longer any tissue in the, underneath where that crinoid cilia are.
Yeah, no, and it just reminded me of all these discussions we've been having about some of these unbranched corals that are sometimes branched, and maybe crinoids or other animals could facilitate that by clearing this little section, and then something could settle on it.
Exactly. I'm so glad you said that. That's precisely what I was thinking. You know, when we were having these discussions on other dives, and we speculated that perhaps there's something that damages the tissues, exposes part of the skeleton, and then as the tissue grows back, perhaps it starts a new growth axis that results in a branch. So clearly the need here is to dislodge that crinoid and then come back in 30 years and see if there's a branch.
That's quite an experiment, yeah. All right.
I think Sirius is starting to come over, you know. Even a little.
Let's zoom out. Sorry, I couldn't keep it more steady there.
But yeah, and then if we have a quick second before we have to move, there's a coral just bottom written out of our view.
If Copilot comes up, you got 30 seconds for you, so he'd give you some room.
Yeah, let's come up.
A lot longer, I'd say.
So it's the right, and it's out of view now. It's just about to coming in view.
Okay. The one there on the side of the rock?
The puffy one straight
dead
ahead.
center now, top of the rock.
Okay.
The lasers are on it.
The one that I was trying to avoid hitting earlier. Okay, zoom in.
Very large polyps on the top.
Not sure that's the same thing as the other one we were looking at. I don't know, Scott, if you want to weigh in on that.
I do not know, Chris. That's why I wanted to get the close-up so that we could compare, and at least now we have this really nice image. What we can do, or what will be done probably by Les or Steve Cairns, is to look at the length of the main axis and see how frequently you're getting that little sort of zig and zag of the skeleton, where the branches are coming out, how many polyps there are per branch. Characteristics like that will be used to help us answer that very question.
Video's clear.
Yeah, but it is interesting that these chrysogorgid corals are quite common, and they're not as pronounced. They don't
Okay, I'll push up. And that heading
jump out at you immediately because they're so wispy and fine.
Same.
But every time we start zooming in, they start popping out. It's a nice view here of the abundance of life. Again, this is quite deep. We are almost 2.5 miles deep.
Not a ton of fish down here, huh?
Yeah, and that's
I think we just went over a white.
I've seen this kind of quite a bit on these
coral
dives,
that was one of those
places where we see the corals and sponges and other invertebrates. The fish are not there, and then when you come to patches where there's not no corals and sponges, you see a few more fish, and that seems to be kind of a general trend.
Science, did you guys happen to notice that white mushroom coral?
I do not.
It was just lower and back where we were coming from, about 30, 40 seconds ago. I also just saw a white squat lobster.
Quick zoom on, just to practice.
Copy that.
And that's exactly what I was pointing out, is that as soon as you zoom in, there's another one of those chrysogorgid corals in the background. very wispy and then thin, but they seem to be quite abundant. But they don't not as pronounced if you're like zoomed out.
Okay, let's zoom out, Roland.
Hey Daniel, we think there are 3 different chrysogorgid corals today on this dive. So this may be a Pleurogorgia, and this is different from the last one we just zoomed in on.
Yeah, I agree. There is at least 3 different forms. And I think there's a lobster.
Yeah,
Let's get.
take a look at him.
And it looks like a squat lobster. And unfortunately, Mary Wixton couldn't join us today, but I'm sure she'll appreciate the images later. She's our expert on many different groups of arthropods from Texas A&M University.
So Dan, as I start moving, do you want me to keep you in view or stay on?
Oh, okay. Well, after this, we'll pick up and go. I'll push out ahead. Just keep me in view for now.
Okay, I'm fine either way.
I'll just be quick here. Go ahead, Roland.
Don't let me rush you. I'm not trying to rush you.
No. I appreciate the heads up. Wasn't sure.
I'll do that for you. I'll do the rushing.
It's partial.
So yeah, that is in the genus Munidopsis, but that's as far as I can go here.
Okay, is that a...
Yeah,
is that good?
Yeah, excellent, yeah.
Okay, let's zoom out.
And there's a question here about the yellow
Get
eyes.
back on heading.
And
I'm not sure we can go further than Munidopsis on this one. He has a very stubby spine coming out between his eyes, and we have a bunch of those in the new OER gallery that we just went to Munidopsis, or shall I say Mary Wickstein just went to Munidopsis.
Yeah, and I was looking through those same images, Chris, and yeah, that's... And there was a question about the yellow eyes, and yeah, in that same guide, there are some that have yellow eyes, but there's also some that do not, so that hopefully provides some context for you, Tina. Yeah, for folks that joined us later in the dive, we are about an hour and a half into this dive, and we collected one geological sample and one biological sample. The biological sample,
maybe come up a few meters.
Chrysogorgid coral. And on today's dive, we have about an hour and a half remaining of bottom time. So because we're so deep, we'll have to come off the bottom at 15 minutes past 2 o'clock Hawaii time.
All right, Chris, we're going to switch out at 1 in 15 minutes.
And then, yeah,
Josh is going to come in 45, so you should just take it.
Maybe we can take a
45 minutes. Sorry.
Quick look at one of these sponges to our right whenever we have
a lot of blue water at the end.
You take it so Josh can get some.
Science wants a few sponges on the right.
Yeah, I'm sorry, Washley, can you repeat that last?
Yeah, one of those sponges to the right, if we can get a zoom.
Sure.
So these are some deeper forms of those sponges we were seeing a lot of yesterday in the genus Poliopogon family, Pheronematidae. And I'll just say a few things about how you can spot these guys. If you see how they're attached to the rocks, they're not glued to the rocks like a lot of the other sponges, the Caliphacus, for example. They actually have these hairy modified spicules or threads that you can see at the base that they're clinging to the rock on, and that's one of the diagnostic features for this group.
Okay, we can go partial.
Yeah, thanks,
Partial.
Chris. And then it's also these atrial surfaces, these flat surfaces, and the amount of them that
Okay, we can go in more.
differentiates for different groups, but a lot of them are just have placeholder names right now. We've had like species A, B, C, D, F, and a lot of these need to be named. A lot of this here in the deep sea, the species, the taxonomy is still needs to be even a framework, and some of the coarser taxonomy and some of the groups needs to be put in place.
So I'm going to use this as a launch pad to talk a little bit about spicules on these guys. The really fascinating thing about these glass sponges is their architecture, their microscopic architecture. And when you actually take a look at some of these sponges, they have all types of different microscopic structures that are just beautiful and fascinating. This group, the family Pheronematidae, is in an order called Amphidiscophora, and it's because they have a special type of spicule called an amphidisc, which looks like two umbrellas that are hooked handle to handle in opposite directions. And it's a really marvelous, beautiful spicule that they have. And what they use that for, I have no clue.
Why don't we have the upper swing arms out?
Okay. Yeah, excellent. Thanks, Chris. And, yeah, definitely, it's very remarkable animals, and some of them are enormous in size.
Zoom in real quick.
And a lot of times when you zoom in on these, you'll see all kinds of other critters on them. You'll see brittle stars, amphipods, polychaetes, shrimp, all kind of other organisms living in that loose texture.
So, Chris, I was curious, what power of magnification do you have to go to before you start seeing those microstructures?
Well, the spicules come in all different sizes, so in some cases you can see them by naked eye, particularly these long thread-like ones that they're attaching to the seafloor. They're individual spicules, so they're quite obvious.
Oh, look at that little gastropod
that I was talking about. They're really small. You're going to need to use probably a 20x microscope to see them.
Yeah, and then that's what I was talking about. We have a gastropod, they're a mollusk in view, so yeah, these sponges provide a lot of habitat, and then even in the internal parts of sponges, a lot of times there's all kinds of animals living inside.
That little guy was cruising.
Okay. Sorry, I'm starting to run a bit.
There's another one of those little spinning guys.
Yeah, quite remarkable animals.
So sponge identification 101, look at how they're attached, and you can eliminate a lot of candidates just by noticing that one feature.
Yeah, and so those that way they're attached to the substrate, that's characteristic of that whole family of sponges, Pheronematidae.
Okay, let's zoom out. Push ahead.
Those structures kind of remind me of the spun glass that comes off of the volcanoes over in Hawaii called Pele's hair. The golden little tiny glass pieces, and I'm assuming almost made up and looking the same way. So the glass that gets spun off of the little bubbles that are popping at the volcanic vents in Hawaii produce a silicon volcanic glass. And Chris, isn't that the same makeup for the sponge's skeleton?
Feeling okay, Pop?
Silicon oxide?
Okay.
I don't know if it's silicon oxide. It's siliceous, definitely, and that's why these are in a group called glass sponges, the Hexactinellida.
Yeah, it looks like we're seeing more pillow lava types of structures now. The geology seems to be changing.
Yeah. I'm just quite amazed at how many animals we do see here. We're now at 3,940 meters, so the peak of this seamount is just below 3,900, about 3,880 is the shallowest we saw on the multi-beam. So this ridge is a very, very gentle slope.
To the left of the lasers is a very small stick. It's actually right between the lasers of coral. If I can zoom in.
Yeah, but we still... we're long since stopped the ship, right?
Yeah, correct. We can get another one in.
To the end of the lasers.
I would think.
Yeah, I think so. What are you, 80? Looks like that's about right for sonar in the distance there. Yeah, let's get another one in. Bridge now.
Okay, go ahead, roll.
It's the black
hey, another
stick.
ship move, please.
To the right.
Same as last. 30 meters, 085 degrees.
Oh, yeah.
0
Yeah, that'd be a good.
2 knots.
Both of them.
So yeah, there's
Good copy. Thank you, Bridge.
a dead coral, and I think we see several gooseneck barnacles on top. Those white objects. Looks like that one might be.
Daniel, was that a cladorized sponge, kind of like a little comb that was on the left? Is that a frame now?
Yeah, I did not see that, Scott.
It's very small. It's next to that Rittigorgia skeleton you're looking at, and it's just come back into view.
Oh yeah, I see that. So the very
left side.
So yeah, left of the lasers, about 2 feet left of the lasers.
Okay.
It's a partial.
Thank you, Bridge.
Good eye, Scott.
Okay, go ahead,
Move
roll.
up closer, I get a better view. I don't know if I'm close enough.
No, if we need to, we can move in closer.
That's max.
How's that, Dan?
Looks like a little black coral.
Yeah, I think so. Yeah, I think we're looking.
That's a black coral. That's an unbranched, or so far unbranched black coral. You can see the nematocysts and the tentacles are right there, and the polyps are really stretched out.
Pulling out.
Yeah, and quite remarkable. And from the polyps I saw, they were very elongate and transversely, and so that would suggest that this is a family Schizopathidae. But yeah, it's unclear if this is just a young one that hasn't yet branched. Yeah, and that's exactly what Scott is suggesting, and that is very interesting. So yeah, these are a different order of corals than what we've seen so far, or most of what we've seen. So these are the black corals, named after the color of the skeleton. It's also one of these groups that's very little known. There's about 250 species recognized right now in 7 families, and there's a lot more species that are yet to be described. These are typically found in deeper waters. Vast majority found at depths below 50 meters, and then with records as deep as 8,000 meters.